Moving on to classification of enzymes. First, enzymes can be classified based on number of polypeptide chains. Enzymes could be monomeric enzymes. Monomeric enzymes means what? It is made up of single polypeptide chain. For example, ribonuclease. And second type is oligomeric enzyme. Oligomeric enzyme which possess more than one polypeptide chain or the subunit. We can call this as subunit also. So more than one polypeptide chain, if they are there, then we can call those enzymes as oligomeric enzymes. And very good example of oligomeric enzymes is lactate dehydrogenase. This example we can remember. There are many examples like aspartate transcarbamylase, that is also the oligomeric enzyme. Then the third type is multi-enzyme complex. This multi-enzyme complex, they possess specific sites to catalyze different reactions in a sequence. So it's a complex of many enzymes. Multi-enzyme complex, they possess specific sites to catalyze different reactions in a sequence. So it's a complex of many enzymes together. Okay, so the intact complex is functionally active and not the individual unit. Okay, so if they are separated, they may not be active. So it's full complex of the many enzymes. So each enzyme will have a definite active site on it, and maybe one substrate is coming, it is activating this enzyme, then second substrate, whatever is the product, or the second substrate will come and attach to the active site of second enzyme, and so the chain reaction, through this sequence of reactions some biochemical processes are catalyzed. So that is the work of these multi-enzyme complexes. One example, if you want to remember, you can remember the pyruvated hydrogenase fatty acid synthase. This is one example. So you can take this screenshot if you want to remember the examples: ribonuclease, lactate dehydrogenase, and pyruvated hydrogenase fatty acid synthase. Second classification of enzymes is based on activity of the enzyme inside or outside the cell. So based on that, there are intracellular enzymes and extracellular enzymes. Intracellular enzymes means what? They are functional within the cells where they are synthesized, and extracellular enzymes are active outside the cell. Okay, they are active outside the cell. All the digestive enzymes here, all digestive enzymes which we have seen on the first slide, I guess, they belong to this group, that is extracellular enzyme. Now the third classification is based on IUB system of classification. Here the enzymes are divided into 6 major classes. Each class on its own represents the general type of reaction brought about by the enzymes of that particular class. So first is oxidoreductases, transferases, hydrolases, lyases, isomerases and ligases. So you can see here in this particular image, the oxidoreductases, these are involved in oxidation reduction reactions, and you can represent this oxidation reduction reaction in this manner, and examples are also given here. From each type, at least one example you have to remember. Then we have transferases that catalyze the transfer of functional groups. Okay, so functional group from this particular enzyme is transferred to B and this product is formed. So this is with the help of transaminases, transmethylases. Third type is hydrolysis, that bring about hydrolysis of various compounds. Okay, so with the help of water, so maybe water is breaking up and it is been taken up by each of the side. So such hydrolases are lipases, esterases. Then lyases is the fourth class. These are specialized in the addition or removal of water, ammonia, carbon dioxide. Means the small molecules are removed here. So here addition and elimination. So this is broken, and it has been added here. So these are lyases. Aldolases are good example of lyases to remember. Then isomerases, enzymes involved in isomerization reaction where interconversion of isomers is required, like triose phosphate isomerase. Last is ligases. Glutamine synthetase is very good example. So these are the enzymes catalyzing synthetic reactions where two molecules are joined together, and for this reaction you can see here ATP is used. ATP is used matlab from ATP its phosphate group is attached here and ATP will be removed as ADP. ATP mein there are T stands for trino. So 3 phosphate groups are there. So one phosphate group will attach to the enzyme and this is getting converted into ADP, that is diphosphate, adenosine diphosphate. So that's it about the classification of enzymes. Next, factors affecting enzyme activity. First factor is concentration of enzyme, concentration of substrate, effect of temperature, effect of pH, effect of product concentration, effect of activators, effect of light and radiation, and last is effect of time. So very important factor among these to remember is concentration of substrate. Rest all the factors are very small, nothing much to explain, but concentration of substrate is the major part, or will be the major part of your answer if the question is asked on factors affecting enzyme activity. So this particular part I will be covering in the next video. Stay tuned for the latest updates on my channel. Thank you very much. Have a nice day.